Welcome to this video where I give an overview of cell structure. Let's look at this cell, which is a typical eukaryotic cell. It's not a plant cell or a fungal cell because there's no cell wall. First, I'm going to look at organelles. These are membrane-bound structures which are found inside the cell. First, let us start with the nucleus. It contains a double membrane called the nuclear envelope. It has different structures, including the nucleolus, where ribosomes are synthesized, chromatin, which is DNA associated with proteins. Now, chromatin can be seen here, and the first structure I've shown here is a beads on a string appearance, and this will be shown like this. It'll appear like this under an electron microscope. Now these beads are nucleosomes. Nucleosomes we can see here contain histones, and the DNA is wrapped around the histones. The histones here are a series of 8 subunits. It's a histone octamer, and the DNA is wrapped around it, and the whole structure is called a nucleosome. And this structure here is about 10 nanometers wide, so it's fairly uncondensed. Now, nucleosomes or histones are the major protein associated with DNA, but there are also other proteins. Now, this nucleosome can condense, it can wrap around itself to form a more compact structure, which is around 30 nanometers wide. This is condensed chromatin. After this condensed chromatin condenses further, becomes more compact, it can eventually form a chromosome. Now, the nucleolus is where ribosomal RNA is produced, i.e., ribosomal RNA genes are transcribed to make ribosomal RNA. The ribosomal RNA associates with ribosomal proteins to form ribosomes. To facilitate this, substances need to enter the nuclear pore as shown here. So ribosomal proteins, which are synthesized outside the nucleus, will enter the nucleus and they will go to the nucleolus where they will be used to form ribosomes. Nuclear pores will also allow transcription factors to enter the nucleus to alter gene expression. Substances that will leave the nucleus through the nuclear pores include messenger RNA, fully assembled ribosomes, and transfer RNA. Now, there are 2 classes of ribosomes. The free ribosomes, as we can see here, associated or not associated with anything. They are floating around in the cytoplasm. And ribosomes that are associated with the rough endoplasmic reticulum, or rough ER, as shown here. Now, ribosomes are not permanently associated with the rough ER. They can associate and disassociate. Normally, ribosomes start translating messenger RNA as free ribosomes. Once they have synthesized a short peptide which protrudes out of the ribosome, then they will associate with the rough ER and they will continue synthesizing that polypeptide. Now, the polypeptide, if it's synthesized by ribosomes attached to the rough ER, that polypeptide will enter the rough ER here, or it will be stuck in the membrane, so it'll be a transmembrane protein. Now, ribosomes that are associated with the rough ER normally synthesize proteins that are being secreted. Ribosomes that are in the cytoplasm, the free ribosomes, normally synthesize proteins that stay in the cytoplasm. An overview of the rough endoplasmic reticulum. So as well as being involved in protein synthesis, rough ER is also involved in protein folding, where chaperone proteins inside the rough ER help proteins to fold once they have been synthesized. Also in protein glycosylation, and this is a post-translational modification. So when proteins are synthesized, if they remain in the ER, for example, in the lumen of the ER, they can be glycosylated, sugar units can be attached. Now, vesicles containing proteins can bud off the ER and move or translocate to downstream destinations, and the most likely destination is going to be the Golgi apparatus. It's important to understand, as I mentioned before, that proteins that are synthesized on the ribosome attached to the rough ER, normally those proteins are going into the lumen of the rough ER. And if that happens, then they are mostly destined for secretion. Sometimes they do end up in certain organelles, as we will see later. Also, some proteins, when they are synthesized at the rough ER, they become transmembrane proteins. They embed in the membrane, they cross the membrane of the rough ER. And these proteins are normally destined to become transmembrane proteins at the plasma membrane. Now, some cells have another type of endoplasmic reticulum called the smooth ER. This is separate from the rough ER. The smooth ER is involved in phospholipid synthesis, steroid synthesis, and lipid synthesis. In most cells, regions of the ER shown here are rough and some regions are smooth, and these regions have no ribosomes attached. In that case, the smooth ER does not really do all of these functions. As I mentioned before, the Golgi apparatus or the Golgi body. This is split into three different sections, the cis, medial, and trans Golgi networks, and we'll see this in a moment. The cis Golgi network, or the CGN, is involved in phosphorylation of oligosaccharides attached to proteins. Removal of mannose residues also occurs here. Now here we've got the Golgi apparatus. You can see the cis region, and this region is going to be the region which is closest or facing the rough ER and the nucleus. The region in the middle, which is the medial region, and the trans region, which is going to be facing the plasma membrane, is going to be the closest to the plasma membrane. So firstly, vesicles from the rough ER fuse with the cis Golgi network or the cis region. Now, there are various theories of how proteins travel through the various membranes of the Golgi apparatus. But eventually, after proteins are processed by the Golgi, they leave the trans region via secretory vesicles. Now, each flattened sac in the Golgi is called a Golgi sac, or the technical term is cisterna. So you can see that the Golgi apparatus is made of many cisternae. Let's look at the medial region, or the MGN. Removal of mannose residues occurs here, as well as addition of N-acetylglucosamine. The trans region, or the TGN, is involved in addition of galactose and sialic acid. Also sulfonation of carbohydrates and tyrosine residues. Okay, let's now move on to the mitochondria. This is the site of some stages of respiration. Remember that glycolysis, the first stage of respiration, occurs in the cytoplasm. After this stage, the next stage is the link reaction, followed by the Krebs cycle and then the electron transport chain. These three steps occur in the mitochondria. You can see the mitochondria is made up of the outer membrane, shown here, and then the inner membrane. The inner membrane is thrown into folds called cristae. This increases the surface area, and it's on this inner membrane that the electron transport chain is found. Proteins of the electron transport chain allow oxidative phosphorylation. The matrix, which is the space inside as shown here, is the site of the Krebs cycle. Another organelle is the lysosome. And the lysosome is involved in breakdown of certain materials in the cell. So if materials are taken up by the cell via endocytosis, then the lysosome is involved in digestion of those materials. So for example, if I have the plasma membrane here, and let's say there's a substance outside, could be a pathogen, that plasma membrane then takes up that substance, forming a vesicle. Here's a vesicle, and you can see that the substance is inside. That vesicle can fuse with the lysosome, so these two can fuse together. Now, the lysosome contains lytic enzymes or digestive enzymes, and they are kept in the lysosome so that they do not damage any other part of the cell. So when this vesicle containing this substance taken up by endocytosis, when it fuses with the lysosome, then you will see that that material will go into the lysosome and it will be degraded. We can also have digestion of cell components, including organelles that have been damaged. In this way, the lysosome will just focus on those organelles and destroy them by digesting them, and then it will recycle those materials, and they'll be used to make new organelles. Another important factor, which is important in destroying cells that are damaged beyond repair, is apoptosis or programmed cell death. Here, the lysosome is involved in destruction of the whole cell. The peroxisome is another important organelle within the cell. It's involved in breakdown of long-chain fatty acids into shorter chains, which can then be respired or used as a respiratory substrate. The peroxisome is also involved in the synthesis of bile acid, an important component of bile, which is necessary for digestion. Removal of excess hydrogen peroxide, which is a substance that builds up in the cell, is carried out by the peroxisome. Too much of this substance will damage the cell. So here, hydrogen peroxide, or H2O2, is broken down into water, or H2O, and oxygen, or O2. And this is a very, very important process to remove this reactive species, hydrogen peroxide, which is an oxidizing agent. And this is catalyzed by the enzyme catalase, which is found within the peroxisome. Other functions of the peroxisome include oxidizing toxins to remove them, and hydrogen peroxide is the oxidizing agent here. So, for example, formaldehyde and phenols, which are found in things like cigarettes, and also alcohols, are things that are oxidized in the peroxisome to remove these toxins from the body. Let's now turn to other structural proteins which are not organelles. So here we have microtubules, and they provide the cell with structure and shape. They are also involved in the translocation or the movement of vesicles and organelles, which can travel along the microtubules. An example of microtubules is the mitotic spindle, and this is something that pulls chromosomes or sister chromatids apart during cell division in stages like mitosis and meiosis. Let's look at a microtubule. It's made up of two subunits, alpha tubulin and beta tubulin, and you can see that they are repeated throughout in this structure. So we have layers. We have layers. Firstly, if you look at the minus end here, we start off with a layer of alpha tubulin, and then beta, and then alpha, and they alternate. And on the other end, the plus end, we end up with a layer of beta tubulin. So here, we need to understand that when cell division occurs, the minus end is going to be the end which is going to be attached to something called the centrosome at the poles of the cell. And the plus end is going to be attached to the centromere via the kinetochore. So it's attached to the chromosome. So let's look at the centrosome here. This is an example of an MTOC, or a microtubule organizing center. This is the site where alpha and beta tubulin attach and nucleate to assemble the microtubule, and it organizes a spindle fiber. Microfilaments are a different class of structural protein. They contain actin subunits. They are involved in cell movement, for example, amoeba that will move towards prey and engulf prey. It will also be involved in muscle contraction. That's a very important component for contraction of striated, cardiac, and skeletal muscle. Here is a structure of a microfilament actin, and you can see that you have many repeating units of actin to form this microfilament. Let's look at another type of filament called an intermediate filament. This is larger in diameter than microfilaments. Examples of intermediate filaments include keratin filaments, an important structural component of hair and skin. Another example is lamins, and these are proteins that form a layer right at the base or right underneath the inner nuclear membrane here, called the nuclear lamina. And this offers structure and support to the nuclear membrane. Now, here, if the nuclear lamina is disassembled, this will cause disassembly of the nuclear envelope, an important prerequisite for cell division to occur. Another important factor of intermediate filament function is where they can alter transcription regulation inside the nucleus, and this is in response to forces or mechanical cues which can alter the shape of these intermediate filaments. I hope you've enjoyed listening to this overview of the cell. Please also look at my other videos on degree level biology.